Good morning, all of you. Good morning. Is my voice clear? Audible? Come on, could you please reply? Make sure that the things are going well there. Okay? Shall we begin? Yes. Thank you. Then, see, up to the last class, I think I was talking about inclusion bodies. Inclusion bodies. Am I correct? Let me recall the inclusion bodies. See, inclusion bodies are non-membranous storage granules. Am I correct? Okay. Non-membranous storage granules. In that, the non-membranous storage granules, mainly they are responsible for just storage of certain kind of food material. In that first one, if you talk about phosphate granules, they store phosphate. Some granules store glycogen. Bacteria store food in the form of glycogen. Keep it in mind. Okay. And some bacteria, so-called blue-green algae, it will store food in the form of cyanophycean granules. Cyanophycean granules. These are all our food storage. One thing is common all of them, that they do not have any sort of membrane. Non-membranous, just simply storage granules found in cytoplasm of prokaryotic cell. We have to call them as inclusion bodies. Okay. Some variety of inclusion bodies are found. They are called gas vacuoles. What is the importance of the gas vacuoles? They are air-filled sacs. Where do you find these gas vacuoles? Simply in photosynthetic bacteria. In fact, in photosynthetic bacteria, these air-filled sacs will make the bacteria to float on the surface of water. If a bacteria submerged under the water, that cannot get sufficient amount of light for the photosynthesis. So, if a bacteria ready to perform photosynthesis, always it has to expose to sun. That's the reason why to float on the surface of water, some bacteria developed some special kind of structures known as gas vacuoles. That phenomena is known as buoyancy or floating on the surface of water. Up to this last class we had this discussion. Now I am beginning the story of eukaryotic cell. You know, eukaryotic cell from the kingdom Protista to Animalia are known as eukaryotic cell. These eukaryotic cell, one interesting property, you cannot comparatively analyze one eukaryotic cell to another. Kingdom to kingdom, structural orientation will be changed. But prokaryotic cells, almost all cells are having same kind of structural orientation. With a slight modifications, they do have some changes, but almost all prokaryotic cell, they are structurally similar. But eukaryotic cell, you can't find their structural similarity. They have some similarities, of course. That's why we have made them a kingdom in the name of eukarya, or in a domain in the name of eukarya. But eukaryotic cell to cell varies. Some cells will have cell wall, some may not have cell wall, some are having centriole, some are having chloroplast, some are having vacuole, some may not have, like that. Kingdom to kingdom, some exclusive properties are going to be added to the eukaryotic cell. Some of the properties we will comparatively discuss. First we will begin with something called plasma membrane. When I had started the discussion of cell, something I told about plasma membrane. Do you remember? If there is a cell, first and foremost thing, it must be having an outer boundary. That outer boundary should be the plasma membrane. Often we get plasma membrane, cell membrane. Which is correct? Both are correct. Cell membrane, plasma membrane. Of course, both are correct. But why do you have one person must be having two names? One person, two Aadhaar cards not valid. But here you have given for one component two names. Why? It is actually cell membrane. Cell membrane. It is in the state of plasma. That's why the name has given called plasma membrane. In that case, you should know one thing. Cell has membrane, of course. That membrane, plasma in state. That's why they call it as plasma membrane. What is this plasma state? It is neither a liquid nor a solid. In between the liquid and solid state, it is revolving. That's why it is called as plasma membrane. One more thing. Wherever you come across a word called membrane, inside the cell, around the cell, that membrane is plasma membrane only. Why I am saying this? You come across endoplasmic reticulum, Golgi complex, lysosomes, vacuoles, many kind of cell organelles we will get randomly. Okay, one by one we will learn. But wherever I say this body possesses something called membrane, you should know they are talking about plasma membrane. Endoplasmic reticulum made up of membrane, Golgi complex made up of membrane, lysosomes have membrane, vacuoles have membrane. What is membrane? That is nothing but plasma membrane that we are going to discuss now. First one. When did you study plasma membrane? Plasma membrane studies, I am talking PM here. PM stands for plasma membrane, not our PM. PM stands for plasma membrane. Its structure deduced after advent of electron microscope. After the advent of electron microscope. Understand that word perfectly. Plasma membrane had been noticed when they were discovered a concept called cell. I think you remember in the cell theory, there were two people who were working, Sleden, Schwann. The Schwann was the first person mentioned. There is an outer boundary present around the animal cell. That outer boundary, right now you are calling it as plasma membrane. Do you remember? But on those days, plasma membrane structure was unnoticeable because its minute structure, plasma in nature, and it could not be easy to recognize. That's the reason why the structural orientation of plasma membrane had been deduced. Deduced means concluded. Correct meaning of deduction means concluded. Deduction means not reduce. There's another synonymous meaning will be there, but deduced means concluded. Concluded after the advent of electron microscope. Advent, this is another English good word. Remember, advent means popularized or in use. For example, before something you purchase, they give advertisement. Advertisement is introduction. Once you purchase that object, you will become advent to it. Advent means that will be used for you. To understand? Of simple, this word if I want to say in a normal, comfortable English, I will say plasma membrane structure concluded after Electron microscope has become the popular in the field. That's what they want to write. So simple sentence, good word. Deduce and advent. These two words remember, future purpose. Next one. Yes, I have the electron microscope and I wanted to use the electron microscope to understand the plasma membrane structure. Excellent, extraordinary. But for that, I must be having some kind of plasma membrane to kept under the electron microscope to watch. That's why model for plasma membrane or model membrane is RBC membrane, which means whatever the data that we have deduced about plasma membrane, this data had given based on the RBC plasma membrane. What does it mean by that? We want one model membrane to study. That membrane had taken from RBC. Why did you go only to RBC? You have thousands of cells in the world or in the nature. Why did you choose RBC? That was an interesting story. RBC present as a part of liquid connecting tissue. Have you heard this name? Liquid connecting tissue? That is nothing but blood. Why did you call blood as a liquid connecting tissue? It is composed of cells, but intercellular cementing substance called collagen is absent. Since collagen is absent, blood has become liquid in state. It has many cells, but they are not interconnected with one and another. That made them individually present but collectively working. Extraordinary nature of blood. Cells present individually, but they are working collectively. In that blood, one popular cell called RBC. This RBC has no nucleus, nothing inside apart from hemoglobin. Take an RBC, make a hole, remove that hemoglobin from inside, then you will get an empty plasma membrane. That empty plasma membrane RBC has a name called RBC ghost. RBC ghost. Sometimes ghost also helping for some studies. The empty plasma membrane of RBC is known as RBC ghost. Then come back. I wanted to study about chemical composition of plasma membrane. Chemical composition. Mainly or majorly lipids. Majorly they are made up of lipids. In addition to lipids, there are proteins, there are carbohydrates, etc. Some other substances will also be present, but major composition is lipid. Now, I would like to clarify one thing about this composition. Composition varies from cell to cell. Composition varies from cell to cell. Variety of cells will have variety of composition, which means percentage or relate to their composition varies. Some will have lipids in high quantity, some may have protein in high quantity, etc. etc. Compositions may going to be vary. But one thing, whenever you talk about plasma membrane, major composition of the plasma membrane always nothing but lipids. But one confusing statement I will write to you. If you take RBC membrane, it has 52% of proteins, 40% of lipids. Something is odd here. Am I right? You said major quantity of the plasma membrane composition is lipid, but you have written in the RBC 52% protein, 40% lipid. Why? Why you generated conflict here? This is not a conflict. Confirming the statement like this. Whenever chemical composition you choose, that chemical composition definitely vary from cell to cell. One more thing I will tell you, there is a membrane called axonal membrane. Axon, dendrite, cell body in the neuron. You take the axonal membrane, study the chemical composition. Around 75% you will get lipids. Very few amount of proteins will be there. They do have proteins, of course, but major quantity of membrane filled with lipids, because on the axonal membrane something is present in the form of myelinated sheath. Myelinated sheath made up of sphingomyelins, a special variety of lipids exclusive for development of Ranvier nodes and also myelinated sheath on the top of axonal membrane. That's why chemical composition will be vary from membrane to membrane. Never mistake, lipids will be lesser in RBC membrane, proteins are more, but it doesn't mean that every plasma membrane lipids are lesser than protein. That would be changeable. Then come back talking about lipids. If you said lipids are the major composition of plasma membrane, I am talking about here lipids. The type of lipids present in the plasma membrane, first variety is phospholipids. This is the major composition. Next one, second quality, cholesterol. Cholesterol also lipid. Cholesterol is a steroid mode of lipid. One thing, cholesterol also there in the plasma membrane, but cholesterol is not universal. Cholesterol for only animal cell plasma membrane, but in our NCERT book, when they are discussing about plasma membrane, here and there they have added the chemical composition cholesterol also. Cholesterol is not universal. Phospholipids only universal, but cholesterol only animal cells. This is interesting thing, ma. Cholesterol never be present in plant cells. I have seen in some advertisement, this is a coconut oil, cholesterol free. This is the advertisement they give. If you simply know some biochemistry, you can understand, you need not to say coconut oil or groundnut oil will have cholesterol, because they are plant products. Cholesterol something that is belongs to only animals. Then the advertisements are always deceiving us. They say it is a sesame oil, cholesterol free. You need not to say it is cholesterol free, I know that, but once it have entered in my body, it may be generate cholesterol. That's a secondary story, but by nature that coconut oil or ground oil or whatever the plant oils that we are getting, definitely they do not have cholesterol. Keep it in mind, in future important. Cholesterol will be absent in case of plant cell membrane. They do have sterols, but they are called phytosterols. That's why often you will see these advertisements, these phytosterols, antioxidants, and they are having some kind of beneficial impacts, etc., etc. Those are all are associated with different types of byproducts that are coming from plant products. Plant oils are always good. That's why there is no supporting of animal oil. Once upon a time in the beginning, whale oil, you know, the sharks, whales living in the oceans, they used to collect them after their death or before their death or by killing them. They have blubber, which is nothing but a thick layer of fat. You know, polar bears, etc., they have thick layer of fat around their body in order to sustain the fluctuations of the food. So they used to collect these whales, and the whale body smashed and slaughtered, collected the fat. From the fat they used to get oils. These oils were that cooking oils or edible oils once upon a time. That was highly dangerous. That's why gradually the animal oil usage decreased and plant oil supported. Now also there are variety of compositions are coming. That's a secondary story. We will discuss about it later. Then right now, confined. Cholesterol is a animal sterol. It could be there in the plasma membrane. Its work is somewhat related. I will talk about it later. First one. When you talk about phospholipids, the type of phospholipid present in the plasma membrane are phosphoglycerolipids. Glycerolipids. These phosphoglycerolipids are important. Phospholipid is a major type. In that phospholipid, one type of lipid present in the plasma membrane known as phosphoglycerolipid. This phosphoglycerolipid contain one glycerol and two fatty acids. One glycerol, two fatty acids. That's why technically it is also known as diglyceride. Diglyceride because one glycerol has two fatty acids. That's called diglycerides. I want you to remember one thing, my dears. Wherever an animal body store fat or lipids, animal bodies store lipids in the form of triglycerides. Animal body need lipids. They will be generally prepared in the form of diglyceride for the sake of membrane and other kind of structural orientation. As you are an animal, you can store lipids. That lipid form that we will store is only triglyceride. You can't store other than triglyceride. In our body, we have adipose tissue and also muscles. Muscles also store triglycerides. Muscles and several other, especially liver also. Several other places we store lipids. Those stored form of lipids are nothing but triglycerides. You want to reduce the weight, reduce your triglycerides. Automatically weight will be reduced. Then come back. Diglyceride story I am talking. In a structural orientation, if you check diglyceride, they appear in the form of head and tail like structure. They have a head, two tails. Never forget two tails. The head. It is made up of glycerol. Because of the glycerol nature, glycerol and phosphoric acid, it is polar. Polar means it has a charge. And also hydrophilic in nature. It can touch to water. Then go to fatty acids. The fatty acids, two fatty acids, this one. Fatty acid. These are long chain non-polar hydrocarbons. Repetitively, carbons will be joined. Non-polar hydrocarbons. These are hydrophilic in nature. Hydrophilic, which means they can, these are hydrophobic in nature. Fatty acids are, of course, hydrophobic in nature. Like this, in one plasma membrane lipid, one part is hydrophilic, another part is hydrophobic. That's the reason why plasma membrane lipids are commonly denoted as amphipathic molecules. Amphipathic. They have hydrophilic head, hydrophobic tail, so that they belongs to amphipathic in nature. I will take these plasma membrane lipids and I am going to arrange in the form of bilayer. Simple. This is a lipid individual structure. In the plasma membrane, it's not that one lipid molecule is present. There are millions and trillions of this lipid molecule will collectively create plasma membrane. How? By forming something that is called as bilayer. Lipids in plasma membrane. Lipids in plasma membrane present in the form of bilayer. They have this physical orientation, bilayer like physical orientation. Why? They wanted to protect their tails from the water. In the cell, in and around both the areas, there is an existence of water. Without water in our body, nothing can be constructed. A cell grows, a cell survives, a cell divides. Wherever there is a cell survival, it must be having water. But the water should not attach to one component of our amphipathic lipid, that is nothing but fatty acid. In order to protect the fatty acid non-polar hydrocarbon tails, there is some special arrangement of lipids developed in our cells, that's called bilayer. What is this bilayer? All the heads okay. Now is it okay, boys? Come on. Done. Am I audible? Yes. Let us see what I was talking. The bilayer has been formed. In the bilayer, always head exposed to aqueous environment. Okay? Always exposed to aqueous environment. Tails are buried inside. Why they have behaved like that? Simple thing. Tails are hydrophobic in nature. They wanted to protect themselves from aqueous environment. That's why they have buried like that. So this is the way there is a formation of bilayer during the development of plasma membrane. Next one. If you talk about plasma membrane, does the plasma membrane possess only our lipids? They do have proteins. Then where are the proteins? Now I am arranging the proteins. Proteins will be present. Some proteins partially buried, some proteins present across the plasma membrane. This is also another variety of protein. Okay. Another variety of proteins, they will be present on the plasma membrane, either attached external surfaces or they may be attached to internal surfaces. These are all our proteins. But one more variety of proteins are there. They are called transversive proteins. They are somewhat bulky, but these are somewhat linear. They are called transversive proteins. The transversive proteins, they will be attached to some amount of carbohydrates. You said carbohydrates are also present. Some amount of carbohydrates, which could be called as sugars. This is sugar. Now come back to the story. I am talking about proteins. Listen. When we start discussing about proteins, about plasma membrane, there are different models are there to elaborate protein. Okay. In that case, in our NCERT book, intelligently they have touched one object only, that is based on ease of extraction. Based on what? Ease of extractions. Based on ease of extraction, that means how easily you can separate these proteins based on some physical forces. Proteins are of two types. Protein. One is integral protein. Another one is peripheral protein, extrinsic. Peripheral. Integral, extrinsic means peripheral. What about integral protein? They are either partially or completely present in the membrane or completely buried in the membrane. Either case, you can choose this. This is completely not buried. Some part is exposed. Some of the proteins may even completely immersed inside the plasma membrane. They are called integral. Peripheral. Peripheral, present externally. They are not completely present. In that case, if I take this plasma membrane, shake it thoroughly, that shake can make these proteins to be fall down. Such kind of proteins which could be easily separated from the plasma membrane are known as peripheral proteins. Next one. There are some proteins which are completely immersed inside the plasma membrane. They are very difficult to separate. If you separated those proteins known as integral protein, plasma membrane structural orientation completely damaged. That's why unless you damage the plasma membrane, you cannot separate the integral proteins. Without damaging the plasma membrane, peripheral proteins can be easily separated. Apart from that, don't touch extra thing about proteins, because about proteins endless information is there on our plasma membrane. Next, go to sugars. One thing, whenever you see the sugar on the plasma membrane, sugars present only external surfaces. External, that means outer side of the cell. Outer side. What does it mean by outer side of the cell? Simple. I have a cell which has this plasma membrane. This is the plasma membrane of a cell. Okay. Then in this plasma membrane of the cell, inner side area is cytoplasm, outer side area is called exoplasm. Wherever you see these sugars, sugars will be attached to proteins. Very important. Sugars will be attached to proteins. These sugars will attach to protein, but those attachment only on outer side. A sugar will be never ever present inner side. Sugar will be never present in inner side. This is a sugar which would be never present in inner side. Okay, the sugar sometimes is also called polysaccharides, etc., etc. But now, cytoplasmic area never ever carries sugar. So, what is the use of these sugars? Sugars on the plasma membrane act as surface markers. Surface markers. And also antigen signals. Antigen or antigen signals. Best example I will give you about the sugar. You know, blood grouping. Blood grouping, A antigen, B antigen, AB antigen, no antigen. Do you remember? A antigen due to one type of sugar. B antigen due to another kind of sugar, like that. Sugars responsible for acting as cell surface marker. A cell I want to recognize. On the surface of the cell, different types of carbohydrates present. Different types of carbohydrates give different variety of antigenic signaling molecules. This is what we call it as, okay, sugar characters. This is about chemical composition. So far, whatever I have explained, what are the chemical components present in the plasma membrane and what is their exact identity. Next, models for the plasma membrane. In the year 1972, there were two people, Singer, what he sings I don't know, and Nicholson. These two people proposed one model for the plasma membrane that is called fluid mosaic model of plasma membrane. Fluid mosaic model for the plasma membrane. What does it mean by fluid. On the fluid, some kind of mosaic things are there. Mosaic means randomly arranged. Randomly arranged, but they are working. That condition is known as mosaic. Fluid mosaic model has been proposed by Singer and Nicholson to explain the physical and chemical properties of plasma membrane. Before fluid mosaic model, there were two more models were very popular. One was called unit membrane model. Another one is called as bilamellar or trilamellar model. These two models were discarded later because fluid mosaic model that was proposed by these people could be able to explain all the physical properties of plasma membrane. Then what is this model says? According to this model, in a lipid bilayer proteins freely floating. Sometimes it is also analyzed like this, or an analogy present for fluid mosaic model. In a lipid sea, protein icebergs are floating. In a lipid sea, protein icebergs are floating. Or else some people also mention like this: in a container, you take sugar solution. In that solution, gulab jamuns are freely present here and there. Whatever the appearance you will see, that appearance is similar to fluid mosaic model that was proposed by Singer and Nicholson. According to them, okay, plasma membrane is quasi-fluid in nature. Quasi-fluid in nature. Quasi-fluid. Most probably, we understand this one in the name of semi-fluid in nature. Quasi-fluid or semi-fluid in nature. What do you mean by quasi? Quasi means partial. For example, I say one statement to you. It is a kind of quasi-government, which means pseudo-government. It's not perfectly formed, pseudo-government. Between one government, another government coming, one gap will be there. That gap will be filled by quasi-government that ruled by governor. Or else something I am saying pseudo. Pseudo is nothing but not perfect, but work will be done. That is called quasi-fluid in nature or semi-fluid in nature. In that, whatever the quasi-fluid properties that the plasma membrane possess, all these properties are due to lipid. Lipids responsible for quasi-fluid nature of plasma membrane. Who is responsible for quasi-fluid nature of the plasma membrane? Lipids. Lipids are the one which are making our plasma membrane neither a solid nor a complete liquid. It is existing between liquid and solid. That composition they call it as a plasma state, or it is also called quasi-fluid state. Now, simple. If you are saying like this, by changing the chemical composition, is there any properties will change? Of course. If lipid composition increase, fluidity of the plasma membrane also increase. If the lipid concentration decrease, fluidity also decrease. What do you mean by fluidity and quasi-fluidity? If you want to understand some analogy, I will explain. Just take it. One liquid we have, universal one, called water. Water is liquid. Does it possess fluidity? Of course, water only possesses fluidity. Remaining all liquids will have either less than water or more than water. Differences are there in the fluidic nature of different other kind of liquids. Only water, liquid state, that state I will consider it as fluidity. Okay, now to understand this, let me take one experimental verification. There is a swimming pool filled with water. You have jumped. You can swim. No issue. You feel why you are swimming in the water. Next, remove the water from the swimming pool. Fill that swimming pool with oil. Or let us take coconut oil. You jump in the coconut oil. Do you feel the same? Of course not. Earlier, in the water, your movements were very easy, flexible. Now in the oil, your movements are going to be restricted. Why? Earlier my movements are easy, now they are very hard to move, because oil has less fluidity when compared to water. Oil is less fluid when compared to water. For example, I am taking fluidity. Fluidity is going to become less. The less fluidity, we will call it as oils. Oils will have less fluidity. Okay, oils do fluid in nature, but that fluidity, when it compared to the water, is less in nature. Next one, let us remove oil from the swimming pool. Fill the swimming pool with petrol, or let us take alcohol. Fill it with alcohol, pure alcohol I am talking. Now jump. The resistance provided by alcohol would be lesser than water. Why? Alcohol is more fluid than water. I told you, the standardization of fluidity we will take with respect to water. Water is a standard measurement for fluidity. Anything that is greater than water, that means fluidity for any kind of liquid that is greater than water, that should be more fluid, more fluid, more fluid, more fluid. They ended with vapor. Less fluid, less fluid, less fluid, less fluid. You will end with solid. Am I correct? I have a liquid and I am reducing its liquidity or fluidity. If I am reducing, where will I end? I will end with solid. If I decrease fluidity, where I will end? I will end with gas. Simple. So I have water which is fluid. That water fluidity, I will take it as standard measurement. Now come back to our plasma membrane. Does the plasma membrane is more fluid than water or less fluid than water? Plasma, I want to answer. Plasma membrane is more fluid than water or less fluid than water? Of course, less fluid than water. That's why they call it as quasi-fluid in nature. Semi-fluid, quasi-fluid in nature. Is this word clear? That's why whenever you talk about the fluidity of the plasma membrane, the fluidity of the plasma membrane is changing gradually. If I want to make the plasma membrane to come near to water, what I have to add? I have to add more number of lipids. If I want to make the plasma membrane to get more rigid or more solid in nature, what I have to add? I have to decrease lipid composition. Clear? Now, lipid increase, fluidity increase, lipid decrease, fluidity decrease. So, one more thing. You are talking about fluid mosaic model and you are saying lipids are responsible for fluidity of plasma membrane. Is there any way that we can measure Fluidity of the plasma membrane, of course, we can measure. They have given the concept like this. The fluidity of the plasma membrane measured as the lateral movement of proteins. Lateral movements of proteins. Fluidity of the plasma membrane measured as a measurement of lateral movement of proteins. You have to understand this perfectly like this. You said earlier, lipid C contain protein icebergs. Most of the area covered by lipids, here and there some protein icebergs are present. If lipids have become more fluid or the plasma membrane composition is more fluid, the iceberg will move easily from one surface to another area. Am I correct? Next, if the lipid quantity is less or fluidity is less, then plasma membrane proteins could not able to move from one area to another area very easily. That's the reason why if proteins present on the plasma membrane could easily move, that thing we call it as more fluid membrane. If the proteins in the plasma membrane moving laterally very hardly, that membrane is a less fluid membrane. In this criteria, let us take one idea for us. You know one thing, take a bucket of water. In that surface area must be more. Take a plastic water bottle, fill that water bottle half with water and throw that water bottle in the bucket. Does it sink? No, it will float. As much as the water you have filled in that bottle, that much water will be good, that much water will sink, and the remaining bottle will be surface. You can see this differences. Now, after few minutes, if you leave that bottle in the center of the bucket, does that bottle will be present forever in the center? No, it started to move different areas. That is called lateral movement. That lateral movement, if it is easily happening, more fluid in nature. Take the same bottle and throw that bottle in oil. Does the movement will be easy or harder? Of course harder. That's what they are defining. The lateral movement of the plasma membrane, lateral movement of the proteins in the plasma membrane is the measurement of fluidity of the plasma membrane. If the proteins present in the plasma membrane could be easily moved, that we will call it as more fluid membrane. Proteins present in the plasma membrane are hard to move. That is called less fluid plasma membrane. Got it? Then what is the use of cholesterol in the plasma membrane? Here, cholesterol, if present, cholesterol act as buffer to the fluidity of plasma membrane. Cholesterol act as buffer to the fluidity of plasma membrane. What does it mean by this? Okay, fluidity of plasma membrane will be having some buffer. For example, in some place you have been exposed to more the melting nature of plasma membrane, because plasma membrane made up of lipids. At the time, you must have some kind of chemicals in the plasma membrane which will reduce that melting. That is nothing but cholesterol. Or else you have exposed the plasma membrane to ice cold environment. Plasma membrane started to solidify. That solidification must be decreased. That is also nothing but our plasma membrane. Fluidity will be having some buffering action. That buffering action is done by cholesterol. If cholesterol present, it will do that action. If cholesterol is absent, sometimes it is going to have some kind of other chemical to maintain this buffering action. But buffering action will be there for plasma membrane. Next one. After that fluidity, let us talk about physical properties of plasma membrane. Simple. Plasma membrane involved in transportation across it. This transportation could be passive transport or it may be active transport. Active transport. First, passive transport is also commonly known as diffusion. Diffusion. Diffusion means, for example, across the membrane, particularly we are talking, okay, across the membrane from high concentration of a substance to low concentration. High concentration of a substance to low concentration. That's called passive transport. Now, first let me complete passive transport, then we will go further. In that case, when I talk about passive transport, so-called diffusion, the passive transport, hereafter I will use a term called diffusion. Listen, this is the plasma membrane, and across the plasma membrane, a material directly transferring from its high concentration to low concentration. This mode of diffusion, we call it as simple diffusion. What is it? Simple diffusion. Simple diffusion. What type of materials could be diffused simply? What type of material could be moving across the nothing happen here, directly one area to another area, this material is moving. That I am calling it as simple diffusion. Do all materials perform simple diffusion? No. Plasma membrane has selective permeability, extraordinary nature that the plasma membrane has to control the movement of the substances across it is selective permeability. But you are saying some materials can be easily diffused across the plasma membrane. So what type of material could be diffused across the membrane easily? They must be small molecule. That molecule should be first small, minute. Why? If it is a large, it will go and hit the plasma membrane and the lipid orientation will be changed. Then ultimately the plasma membrane will damage. So molecule which undergo simple diffusion must be small. Next one, uncharged. Uncharged. Charge should not be there. If they are charged, they are active for reaction. They are uncharged. Next one, hydrophobic. Why hydrophobic? Because most of the area of the plasma membrane made up of hydrophobic environment. You can take that molecule which is hydrophobic easily from one side to another side by simple diffusion. This is for normal kinds of another thing. Polar, non-polar, these words, my dear. Polar means charged, non-polar means uncharged. Keep it in mind. In that case, when you talk about simple diffusion, one thing you should not forget. Whatever it may be the gas, whatever it may be the size, gases are generally have small size, of course, over there, always gases, simple diffusion. Any gas you take, either it's an oxygen or carbon dioxide or methane gas, whatever it, if the gas is traveling across the plasma membrane, the gas travel only by means of simple diffusion. Another mode of transportation is not there for gases. Next one. Talk about one more mode of diffusion is there. There is a molecule. This is that molecule, or a compound, or a substance, whatever you like. It wants to travel from one side of the plasma membrane to another side along the concentration gradient. Okay, along the concentration gradient. It's already present in this side, higher concentration, and it wants to move to another side towards lower concentration. Then, while it is moving, it will come across our plasma membrane. Plasma membrane will stop it. When does the plasma membrane will stop it? If this molecule is simple, it is a large charged polar. Next one, hydrophilic. Okay, this molecule, if it is crossing across the plasma membrane will stop it. No, you are not allowed to go. Why? You are large, you are charged, you are hydrophilic molecule. Then should I stop forever here? Then plasma membrane think, and after that it will give an idea. No, you will be permitted. I will give permission to you. Let me generate some facility where you can travel from your high concentration to low concentration. That facility is this area. You want to go only through it. I have generated one facility to you. There only you have to travel. This diffusion is known as facilitated diffusion. Facilitated diffusion. You are diffusing, but diffuse selectively. Diffuse when I provide the facility to you, there only diffuse. Don't diffuse randomly and don't damage the plasma membrane structure. This is called facilitated diffusion. Either you take simple diffusion or facilitated diffusion. In both the cases, diffusion never take energy from the external sources. Plasma membrane is not responsible for making these components to move. They are moving. Plasma membrane has given the path. They are moving by themselves. Be careful. They are moving by themselves. Plasma membrane has given a chance to move through it. Some molecules, let them go. Some molecules facilitatively go. Plasma membrane is not stopping them. Is it okay? This phenomena is called diffusion. But whenever facilitated diffusion happen, it will use some special kind of carrier proteins. These carrier proteins are known as channels. Channels or porins. Special proteins. Do channels take any energy? No. Why should they take energy? They are making this molecule to move from its higher concentration to lower concentration. Just they have given a path. They open the gate. That's it. They did not make this molecule to move. The molecule is moving by itself. Is it okay? Next one. There is another mode of diffusion is there that we should not forget. That mode of diffusion is known as osmosis. Have you heard this name? Osmosis. In Latin, osmo means water. Diffusion of water across plasma membrane. Across plasma membrane is known as osmosis. It's very interesting concept. Osmo. This is the plasma membrane and he is our water. And it wants to move from its higher concentration to lower concentration. Sometimes water directly allowed to move. Sometimes. Okay, because water is a small molecule, its molecular weight is around 18. Enough, go. You are a small molecule. Is it okay? This is simple diffusion. Sometimes water will be allowed to pass. from its higher concentration to lower concentration selectively. Okay, higher to lower. This diffusion is called facilitated diffusion. Either you allow directly by simple diffusion or by facilitated diffusion. This mode of passive transport of water across the plasma membrane should be denoted as osmosis. Careful. Osmosis is a diffusion, but all diffusions are not osmosis. Why? Imagine there is a tank full of water. You have opened the tap, water is coming. This is diffusion. During rainy season, in the river, water is traveling towards the sea. It's a diffusion. And water is getting evaporated. That is also diffusion. But what is osmosis? Osmosis, water will move from higher concentration to lower concentration only across the plasma membrane, which means while water traveling, if that water has crossed plasma membrane, then only you have to call that mode of diffusion is known as osmosis. All diffusions of water, not osmosis, but osmosis is a kind of diffusion. Next, osmosis always a passive process. Osmosis never ever consume energy. In fact, if osmosis can consume energy, okay, you cannot supply that much amount of energy to make the water to move from one place to another place, because 75% of the body filled with water. If the water is itself is taking energy, that much energy you cannot able to, okay, you cannot able to supply. Next, final conclusion, there is a mode of operation called active transport. What do you mean by active transport? Active, from low concentration to high concentration across the membrane with the expenditure of across the plasma membrane with expenditure of energy. That is called as active transport. Simple. Take the plasma membrane. This is the plasma membrane. Okay. In the plasma membrane, this is that molecule present in low concentration this side. This molecule present this side, very low at concentration. This is very low concentration. And this side it is present high concentration. And our interest of traveling here from the lower to high concentration. Low to high. Ask this molecule, go from low to high. It will say no, because that is against to thermodynamic principles. Nothing can be moved by itself from a lower energy to higher energy. Nowhere else it is possible randomly to a molecule to move from its lower energy to higher energy. So this molecule never move. Then what to do? Actually, active transport is an interesting phenomena where molecule is not interested to move. But here in the earlier cases you said these molecules are moving themselves from higher concentration to lower concentration. In their journey they have come across plasma membrane. But here the molecule does not like to move. Who want that molecule to move? Our plasma membrane. Then plasma membrane will make this molecule to move from its lower concentration to higher concentration by the expenditure of energy. It gives energy to a special type of transporter protein known as pump. Its name is pump. That pump will consume energy, take this molecule and make the molecule to move from its lower concentration to higher concentration. This is possible. Simple. Whenever there is a presence of transportation of any substances. That substance, while moving from lower to higher, by itself, whatever you do, it cannot move. But you can make that molecule to move by using a pump. For example, water we wanted to transport to the tank. In our home, there is a borewell from where you wanted to transport water to tank. For that, we use current. Current is the source of energy. Do you think you put that current to water or motor? Of course, to the motor. Motor consumes energy and pumped the water. So finally, you have supplied energy to pump the water from lower to higher. That's what is happening during at the time of active transport. The best example for the active transport is sodium and potassium pump that is present on plasma membrane. Be careful. Where do you think the sodium potassium pump is present in our plasma membrane? What type of plasma membranes are having this sodium potassium pump? What type of membrane is having this sodium potassium pump? Come on. Careful. Every membrane, every part of the cell, the sodium potassium pump will be present. I have seen many students are saying it is present in axonal membrane of neurons. Yes, it is present there. But not only there. Wherever there is a presence of membrane, there it must be present. Otherwise, that membrane cannot act as electrically active membrane. There is something is called resting membrane potential, minus 70 millivolts, responsible for under the standardized condition to receive electric impulse. That resting membrane potential generated by our sodium potassium pump. This about plasma membrane, that much is enough. Then go to cell wall. Cell wall. Cell wall. Okay. Does the cell wall living or dead? It is dead. Why? It has no living properties. Plasma membrane excrete it and make on the top of it some kind of protective covering. That's called dead. Next one. Is it present external or internal to the cytoplasm? External to the cytoplasm. Cell wall is always external to the cytoplasm. Here, cell wall, if I am talking, I will discuss generally about plant cell wall. Plant cell wall. It doesn't mean that only plants will have cell wall. You know that in the 5 kingdom classification, bacteria possess cell wall. Some kind of protista members like chrysophytes, dinoflagellates will have cell wall. Fungi will have cell wall, plants will have cell wall. Of course, animal cells do not have. But when it comes to cell wall in a eukaryotic cell, usually we choose plant cell wall. It doesn't mean that plant cells only will have cell wall. Got it? Next one. The plant cell wall, I will be divided it into 2 categories for my convenience. One is primary wall, primary cell wall. Another one is secondary cell wall. Who is this primary wall? Primary wall is thin, somewhat thinner, thin, elastic, which means expandable, and also gradually diminishes. Gradually diminishes, which means it will disappear gradually. It never be present permanently. It is there in the beginning, gradually it will disappear. Okay? If the primary cell wall present, what is its composition? Cellulose, of course, it is the major one, and also hemicellulose. Okay. Pectin, and rarely some amount of proteins also could be noticeable. primary wall. Next one. Go to secondary wall. Where do we see something called secondary wall? Secondary wall, always secondary cell wall. Its location is interesting area. It is present between plasma membrane and primary wall or primary cell wall. Plasma membrane and primary cell wall. Which one? Secondary cell wall. Then when we talk about the primary wall and the next one is secondary wall, majorly secondary wall majorly made up of lignin. It is made up of what? Lignin. Does possess cellulose, hemicellulose, some other substances will also be there, but in addition to them, lignin is the major component. When I was talking about anatomy, I might have remind this statement to you. Wherever you find a cell with secondary wall, that cell is already died. Dead cells only generally possess secondary walls. Next one. Go to, apart from that, plant cells, algal cell walls. Algal cell wall. Algal cell wall, somewhat, very interesting it is. When we were discussing about kingdom plantae, green algae, red algae, brown algae, if you take care there, green algae, inner layer is cellulose, outer layer is pectin. Brown algae, inner layer is cellulose, outer layer is algin. Red algae, inner layer is cellulose, outer layer is carrageen. Do you remember these names? That's case. When you talk about algal cell wall, chemical composition of algal cell wall quite different from that of plant cell walls. There they have cellulose, galactons, galactose polymers, mannans, and sometimes minerals like calcium carbonates. Minerals like calcium carbonate. That's algal cell wall composition. We will not talk about algal cell wall detail here. Majorly we target about plant cell wall details. Take a plant cell and its cell wall. In this case, observe, I am using, there are two plant cells. This is the plasma membrane of one plant cell. This is the plasma membrane of another plant cell. And they have cell wall. I am writing this is a primary cell wall. This is primary cell wall. Primary cell wall. Okay, these two are plant cells. This is plasma membrane, this is primary cell wall. Where do you think the secondary cell wall will be deposited? Secondary cell wall exactly deposited here, where in between the primary wall, this is the primary wall, and plasma membrane. This is the place where secondary cell wall is going to be deposited. Gradually, layer by layer, secondary cell wall is going to be deposited. Think about that. I am making the secondary cell wall has been deposited. This much ugly it will not be deposited. It will be deposited smoothly, layer by layer. Okay. Then, in this, you have to notice this is plasma membrane. Okay, sorry, this is plasma membrane, this is primary wall, and this one is secondary wall. In this case, The two neighboring cells of a plant tissue, this is one cell, another cell, both are joined by something that is called as an intercellular cementing substance. This material is known as middle lamella. Could you tell me the composition of middle lamella? Middle lamella. The middle lamella, it is made up of calcium pectates. Made up of what? Calcium pectates. It is a glue-like material. It glue the two neighboring cells stay together. That's why the work of middle lamella is exactly similar to the work of collagen in case of animals' tissues. Over there, in some places, these areas will have some gaps. You know, these gaps are known as pits. When I was talking about plant tissues, I might have mentioned the gaps present in the walls are known as pits. See, one cell wall, another cell wall has a pit. Both will be joined by through this pit. This cell cytoplasm, that cell cytoplasm, both will join. That joining place is known as plasmodesmata. There are some connections present between the cytoplasms of two different cells. The connections are known as plasmodesmata. They are not middle. Be careful. Two neighboring cell walls are connected by middle lamella. Two neighboring cell cytoplasms are connected by plasmodesmata. Plasmodesma singular, plasmodesmata plural. Okay, this is about the how two neighboring plant cells are connected together. I think you're patiently listening whatever I am saying. That is appreciating. I have to appreciate your patience. So tell me if you have any doubts, I can able to help you. Come on, post it there. Are you able to follow what I say? Come on, say something. Yes, the carrier proteins in the plasma facilitated diffusion are a variety of integral proteins. Am I covering all the things that are present in your NCERT book? Am I? Most probably. Okay. Middle lamella is also made up of magnesium. Yes, middle lamella made up of calcium and magnesium pectates. But in NCERT book, they have majorly given calcium pectates. That would be noticeable. Okay. Come on. Any more things, my dear? Middle lamella is the first layer formed during the development of cell wall. I will discuss about that word when we will start cell division. That's a cell division topic area. We will cover it up. Next one. Any more, ma? That's fine. Thank you. We'll meet in the next class.